Dear students, welcome to the fourth session of Histology, Animal Behavior and Applied Zoology paper of fourth semester Zoology described by the Mangalore University. In this session, we shall consider the study of histological features of mammalian intestine. In this session, specifically we are referring to the small intestine. The small intestine of mammals, in particular the human beings, is approximately 7 meters long and has three different regions which are categorized based on the function and structure of the intestine. These regions are the proximal duodenum, the middle jejunum, and the distal ileum. The small intestine secretes several hormones. It continues and largely completes the digestion of foodstuff and absorbs the resulting metabolites. The luminal surface of the small intestine possesses plicae circularis, intestinal villi, microvilli, which collectively increase the luminal surface area by a factor of 400 to 600. This increased luminal surface area greatly aids in absorption of the digested food materials. This is the histological denotion of the small intestine. This is a part of transverse section of mammalian intestine. As we all know that the alimentary canal, irrespective of their position, has got four important histological regions. The outermost covering serosa, the second layer called muscularis externa or simply muscularis, the third layer with blood vessels and connective tissue called submucosa, and the innermost layer with specialized functions are called mucosa. So irrespective of the position of the alimentary canal, these four structures, histological structures, remains the same. The only difference is there will be different kinds of adaptations of these four structures based on the function of that particular organ. As in stomach, it mainly has secretory function, but in intestine it has both, the mucosa has both secretory as well as absorptive function. Mucosa of the small intestine. The epithelium of the mucosa of small intestine is made up of simple columnar cells. It is composed of goblet cells, surface absorptive cells, and some DNES cells. Now let us see the structure of goblet cells. Goblet cells are unicellular glands that produce mucinogen, which accumulates in the membrane-bounded granules distending the apical region of the cell. After being released, mucinogen becomes hydrated and is thus converted to mucin, a thick viscous substance that acts as a protective coating of the epithelial lining of the lumen. The goblet cells have their nucleus and other organelles in the basal region of the cell, which is classically considered as stem of the cell. The goblet cells increase in number from duodenum to ileum. This increase in number is because the secretion of goblet cell is more required in ileum and jejunum rather than the duodenum. The second important type of cell that is present in the mucosa of small intestine are surface absorptive cells. These cells are tall columnar cells with numerous mitochondria, smooth endoplasmic reticulum, which is abbreviated as SER, and RER or rough endoplasmic reticulum, and a Golgi complex. These surface absorptive cells possess a layer of closely packed microvilli with striated border on their free apical surface. They have a glycocalyx which overlies the microvilli and binds to various enzymes including disaccharides and dipeptides, and have well developed tight junctions and zonula adherence. The third important kind of cell that is present in the mucosa of the small intestine is DNES cell. This DNES cells produce and secrete certain hormones like gastrins, cholecystokinins, gastric inhibitory peptide, and several other hormones. The next part in the mucosa of the small intestine is lamina propria. So this lamina propria occupies the course of the villi and the interstices between the numerous glands called crypts of Libercal. So this is the enlarged portion of a villi, and this place is called lamina propria. The space inside the villus is called lamina propria, where we can see many neurons, lacteals, capillary networks, etc. This lamina propria consists of loose connective tissue with lymphoid cells, fibroblasts, mast cells, smooth muscle cells, nerve endings, and lymphoid nodules. They also contain certain specialized structures called lacteals, which are blind-ended lymphatic vessels, and some capillary loops. While discussing about villi and other structures of mucosa, we came across a specialized tubular glands called crypts of Libercal. You can see here some structures in the lamina propria which look like a blood vessel or a canal surrounded by small glandular cells. These structures are called crypts of Libercon. Crypts of Libercon are simple tubular glands that extend from the intervillar spaces to the muscularis mucosae of the intestine. They are composed of goblet and oligomucous cells, columnar cells, and these columnar cells are similar to surface absorptive cells, DNES cells, regenerative cells, and certain specialized cells called Punnett cells. What are these Punnett cells? Punnett cells are located at the base of crypts of Libercon and are pyramidal cells that secretes the antibacterial enzyme called lysozyme, stored in large apical membrane-bounded secretory granules. These cells also release other antimicrobial agents, defensins, and tumor necrosis factors, which are also called as TNFs, and display extensive rough endoplasmic reticulum at the base and a large supranuclear Golgi complex. Supranuclear Golgi complex means the Golgi complex which is present over the nucleus, and number of mitochondria as well. Regenerative cells. These are the cells located in the basal half of the crypts of Libercon and are thin, tall, columnar stem cells that divide to replace themselves and the other types of epithelial cells. So these are very important cells as far as the regeneration and wound healing and replacement of other types of cells of the intestines are concerned. Lymphoid nodules are usually small and solitary and are located in the lamina propria of the duodenum and jejunum and generally be absent in the lamina propria of ileum. They increase in size and number in ileum where they form large continuous aggregates known as Peyer's patches which frequently extends through the muscularis. See, the lymphoid nodules are usually small and solitary and located in the lamina propria of duodenum and jejunum. But while reaching the ileum, the number of these lymphoid nodules increase greatly in size and number, leading to the formation of special large continuous aggregates of lymphoid organs called Peyer's patches. The muscularis mucosae is composed of an inner circular layer and an outer longitudinal layer of smooth muscles. So you can see here immediately after the mucosae there is a small layer of two-layered muscularis. This region is called muscularis mucosae, which is made up of an inner circular and outer longitudinal layer of smooth muscle. The next part in the histology of intestine is the submucosa. The submucosa of small intestine consists of fibroelastic connective tissue containing blood and lymphatic vessels, nerve fibers, and Meissner's plexus. They also house certain specialized glands called Brunner's glands, which are present only in the duodenum. These Brunner's glands produce an alkaline fluid and urogastro. The alkaline fluid protects the duodenal epithelium from the acidic chyme, and the urogastrone is a polypeptide hormone. It's also called human epidermal growth factor that enhances the epithelial cell division and inhibits the gastric hydrochloric acid production. So that means the urogastrone on one hand enhance the division of epithelial cells in the duodenum. On the other hand, it inhibits the production of HCl in the stomach. The next part in the histology of small intestine is the muscularis externa. The muscularis externa of the small intestine is composed of two layers of smooth muscle, an inner circular. You can see here these two parts are collectively called as muscularis externa, and the innermost part is made up of the circular and an outer longitudinal layer of smooth muscle. The inner layer participates in the formation of ileocecal sphincter. The orbach plexus is housed between these two layers. So that means in between the two layers of the muscles there is a mesenteric plex, or this plex is also called as orbach plexus. Externally, the small intestine is covered by a special layer called serosa. It covers all of the jejunum and ileum and a part of duodenum. The adventitia covers the remainder part of the duodenum. So that's all about the histology of small intestine. For more information and more explanation, you can refer these textbooks, which are readily available in most of the college libraries. Thank you.